Welcome back to the deep dive. Today we're taking, well, maybe the most extreme shortcut possible to getting really well informed about this complex intersection of genetics, cell biology, and disease. Our source material today, it's derived straight from cutting-edge pathology texts and molecular biology research. It takes us deep inside the fundamental unit of life, the cell itself. We're diving into a specific, and actually collectively quite significant, class of genetic disorders, the Lysosomal Storage Diseases, or LSDs. Now, I know what you might be thinking, okay, rare genetic disorders, maybe they don't affect me directly, but here's why understanding this is absolutely vital, really. LSDs, it turns out, are providing this critical foundation for understanding neurodegeneration. I mean, in the failure points we see in these rare conditions, they are, molecularly speaking, identical to processes driving really common diseases, things that impact millions, like Parkinson's and Alzheimer's. So the knowledge we gain here isn't just about, obscure pathology. It's potentially the future roadmap for preventative medicine.
That's absolutely right. And that's the core of our mission today. We're exploring the devastating cascade that happens when the cell's internal recycling center, the lysosome, fails to do its job. You can think of the lysosome as the ultimate quality control and waste disposal plant, really. And when the garbage collector basically go on strike, toxic, partially degraded material just accumulates. And that triggers widespread cellular damage. It's quite serious. These LSDs, taken together, affect roughly 1 in 5,000 live births. So as a group, they're actually pretty significant. Fundamentally, they result from inherited deficiencies, usually in essential enzymes, sometimes transport proteins that are needed for breaking stuff down for catabolism. Most follow an autosomal recessive pattern. That means, both parents have to be carriers. But we will talk about one key exception later, Hunter syndrome. That one's X-linked. We're really looking at errors in basic foundational breakdown processes, errors that then cascade into these profound systemic problems affecting multiple organs, especially the brain and the liver.
Okay, right. Let's unpack this then, starting right at the beginning. Before we can really talk about failure, we need to appreciate the remarkable setup of the lysosome itself. If the cell is like a busy city, the lysosome isn't just a recycling plant. It's more like a high-security, super efficient digestive silo. Its main job is essentially cellular housekeeping. It maintains this intensely acidic internal environment, right, which is needed for its tools, the acid hydrolases, to work. And these hydrolases, they're like a whole battery of enzymes needed to break down really complex macromolecules. So where does all this incoming waste actually come from?
Good question. It basically comes from two distinct streams. First, you've got stuff coming from outside the cell. That's through a process called heterophagy, where external materials get engulfed. Second, and maybe even more importantly for quality control, it comes from the cell's own turnover, getting rid of old or damaged components. That's autophagy. In both cases, the lysosome is where the final digestion happens, but those enzymes, they're powerful. They can't just be floating around freely in the cytoplasm. So the cell has this incredible, really complex sorting mechanism to make sure they get delivered precisely to that acidic lysosome and absolutely nowhere else.
Right, and this is where that whole address label idea comes in. Because if that label is missing or wrong, the consequences are, well, immediate and pretty disastrous for the cell. So tell us about this sorting mechanism.
Yeah, it's a beautifully orchestrated process. Manufacturing and delivery all rolled into one. Like other proteins that are destined for secretion or membranes, these lysosomal enzymes start their journey in the endoplasmic reticulum, the ER. Then they move to the Golgi apparatus for final processing. And it's in the Golgi that the cell basically sticks on this address label or molecular tag. The specific tag is the attachment of terminal mannose 6 phosphate groups, M6P for short. It gets added to some of the enzyme's sugar side chains, the oligosaccharides. This phosphorylated mannose is the specific universal tag. It marks the enzyme. Destination, lysosome. Think of it like a unique zip code, maybe a tracking number, for internal delivery only.
Got it. So the M6P receptor, which sits on the Golgi membrane, is basically the delivery truck driver who only accepts packages with that specific M6P zip code. So what happens if the enzyme is made correctly, but maybe that M6P tag isn't applied right, or maybe not at all?
Well, that's the classic and really devastating scenario we see in something called eye cell disease or mucolipidosis too. You see, the Golgi membrane, it's studded with these M6P receptors on its inner surface. And these receptors, they recognize and bind only those M6P tagged enzymes. This segregates them from all the other proteins heading to, say, the plasma membrane or going outside the cell. Now, if the enzyme that's responsible for creating the M6P tag is deficient, Well, then all lysosomal enzymes fail to get tagged properly. They can't bind the receptor, they don't get packaged into the right transport vesicles, and instead of going to the lysosome, they end up being secreted outside the cell, misdelivered, essentially.
Wait, hang on. So the cell is actually dumping its vital digestive enzymes out into the extracellular fluid?
Yeah.
That sounds bad.
It is exactly that, and it's very bad. The lysosomes inside the cell are left mostly empty, lacking functional enzymes. This leads to this massive systemic buildup of macromolecules that should have been degraded, hence the name eye cell disease, inclusion cell disease. The pathology is severe. You see coarse facial features, bone deformities, because the accumulated stuff also messes up fibroblast function, affecting connective tissue. The main point here is that this highly specific tagging and sorting system is absolutely fundamental. When even one step goes wrong, like missing just one tagging enzyme in eye cell disease, the entire lysosomal system fails. And that leads us to the core problem in all LSDs.
Okay. And so when one of the actual breakdown enzymes, the catabolic ones, is missing, that's when the whole system breaks down, creating that metabolic block that defines an LSD. The primary consequence seems obvious. Waste builds up. But you mentioned something more complex. A secondary consequence is vicious cycle driving widespread damage. Let's really drill down into that.
Right. So the initial problem is what we call primary accumulation. The specific substrate that the missing enzyme was supposed to break down, well, it remains partially intact or completely intact. This leads to the buildup of these insoluble metabolites, these complex substrates. They physically stuff the lysosomes, making them swell up. But honestly, if the problem stopped right there, these diseases wouldn't be nearly as severe as they often are. The real widespread damage comes from this vicious cycle of secondary storage and toxicity. And this, crucially, is the direct molecular link we see to chronic age-related neurodegeneration. You see, the primary block effectively gums up the entire lysosomal system. And this matters hugely because lysosomes are tightly linked to the machinery of cellular self-eating, which is autophagy.
Ah, okay. So when the cell tries to send its damaged parts, like old organelles, to the recycling center, but the center is already full of this primary waste, the damaged parts just end up sitting there. They can't get processed.
Precisely. That accumulation of undigested primary material drastically slows down the rate at which lysosomes can process anything else delivered by those autophagic vacuoles. This impairment is devastating for cellular quality control, especially for a specific process called mitophagy.
Mitophagy, that's the targeted cleanup of mitochondria, right? The cell's power plants.
Exactly. Mitophagy is the targeted degradation of old, worn-out, or dysfunctional mitochondria. Mitochondria are essential, but if they start failing, they become toxic. They leak reactive molecules. So when mitophagy is impaired because the lysosome is essentially jammed, these dysfunctional, leaky mitochondria persist. They don't get cleared away. These faulty organelles then generate massive amounts of reactive oxygen species, free radicals, which damage everything around them. DNA, proteins, lipids, and what's more, these leaky mitochondria can release molecules like cytochrome C, which directly trigger the cell's self-destruct pathway, apoptosis. So you see how the original lysosomal enzyme deficiency, through this knock-on failure of autophagy, and specifically mitophagy, is causing cellular self-poisoning and pushing cells towards programmed death.
Wow. And if that wasn't bad enough, this impaired quality control system also lets other toxic stuff build up, like aggregate-prone proteins. This sounds like where LSDs really start to intersect with things like Parkinson's and Alzheimer's.
Absolutely. That failed autophagy and protein degradation system leads to a secondary accumulation of abnormal protein aggregates. Our sources specifically mention polyubiquitinated proteins, that's proteins tagged for destruction but not getting destroyed, and aggregate-prone polypeptides, things like alpha-synuclein and Huntington protein. They start accumulating in these dysfunctional lysosomes. And of course, these accumulating proteins are the defining pathological hallmarks of Parkinson's disease and Huntington's disease, respectively.
That molecular insight is just staggering. We're looking at a shared pathology, fundamentally rooted in the same organelle, the lysosome. And the clinical data backs this up strongly, doesn't it? The Gaucher disease carrier state, just being a carrier, is a significant genetic risk factor for Parkinson's. And actual Gaucher patients have something like a 20-fold higher lifetime risk of developing Parkinson's compared to the general population. That's huge. And then Niemann-Pick type C, increasing Alzheimer's risk.
It really connects the dots between the mechanism and the clinical reality. The initial defect in lysosomal function, whether it's an enzyme missing or a transport protein failing, it cascades. It becomes this broad, multifunctional failure involving autophagy, mitochondrial health, immune responses, even membrane repair. It's all interconnected. Now, to understand why different LSDs present with different symptoms, we need to look at organ specificity. Basically, where the stored material tends to accumulate dictates which organs suffer the most damage. For example, the brain. It's uniquely rich in certain complex lipids called gangliosides. So if you have a defect in breaking down gangliosides, like in GM1 or GM2 gangliosidosis, the accumulation happens primarily within the neurons, and that leads to severe progressive neurological symptoms.
Okay, that makes sense.
Yeah.
And conversely, if the material that builds up is something handled more by say, the immune system, then the disease looks more systemic.
Exactly right. Cells of the mononuclear phagocyte system, or MPS, think macrophages and related cells found in the spleen, liver, bone marrow. These cells are packed with lysosomes. It's their job to constantly degrade diverse cellular debris and foreign materials. That's why so many LSDs result in massive enlargement of the spleen, which we call splenomegaly, and the liver, hepatomegaly. It's because these MPS cells just get stuffed absolutely full of undigested substrate.
Right, okay. This provides a great framework. Let's transition now from the general mechanisms to the specifics, the clinical realities of some key diseases, starting with sphingolipidosis. These disorders are rooted in lipid metabolism. Okay, our first big prototype, Tay-Sachs disease. It's the classic GM2 gangliosidosis. For someone listening who's maybe heard the name but doesn't know the details, what's the core molecular problem here? What's accumulating?
Tay-Sachs is caused by a severe deficiency, usually a complete lack, of an enzyme called hexasaminidase A. Specifically, it's the alpha subunit, hexa. This hexa enzyme is absolutely critical for breaking down GM2 gangliosides. Now, these gangliosides are normal components of neuronal membranes. They're needed. But without the enzyme to catabolize them, the GM2 just builds up relentlessly. We really have to mention the specific demographic aspect here. Tay-Sachs has this, well, notorious prevalence in individuals of Ashkenazi Jewish descent. The carrier rate is tragically high in that population. Something like 1 in 30 people carry the gene.
Wow, 1 in 30.
Yeah, and this high carrier frequency is exactly why widespread genetic screening programs are so essential in those communities.
And the pathology, as you mentioned, is just devastating, and it's focused almost entirely on the central nervous system, right? What does that accumulation actually look like inside the brain cells?
Inside the brain, the accumulation is profound, really. Neurons throughout the central nervous system and also in the peripheral autonomic nervous system, they swell up, they become visibly ballooned because their lysosomes are just distended, completely stuffed full of these gangliosides. If a pathologist looks at these cells under an electron microscope, they see something very characteristic, pathognomonic really, these world configurations. Or sometimes they're described as onion skin layers of concentric membranes inside those swollen lysosomes.
Like tiny onions inside the cell.
Kind of, yeah. And this physical distension, this swelling, fundamentally disrupts how the neurons signal and function.
The clinical timeline is it's perhaps the most tragic part. Infants are born,
Yeah.
and they appear completely healthy at first.
Yeah. They do. The onset is insidious. It creeps up, usually starts becoming apparent around 6 months of age. And from that point on, the deterioration is just relentless. Children experience rapid motor and mental regression. They become listless, lose muscle tone, eventually develop blindness, dementia. There's also a key diagnostic feature, although it's not totally unique to Tay-Sachs, but very characteristic. The cherry red spot in the macula of the retina.
Okay, I think we should clarify what that cherry red spot actually is. The name sounds a bit dramatic, maybe misleading.
That's a good point. It's actually an optical illusion, a contrast effect. What happens is the retina's ganglion cells, nerve cells, they get heavily involved. They become massively swollen with the stored gangliosides. This swelling makes the surrounding retina look pale, almost whitish. However, the macula, the very center of vision, naturally lacks these ganglion cells. So in that central area, the underlying tissue, the choroid, which is rich in blood vessels and normally looks red, is visible. The stark contrast between the pale, swollen surrounding retina and the normal red appearance of the macular choroid creates that striking cherry red spot appearance.
Ah, okay, so it's normal tissue showing through against a diseased background.
Exactly. And due to the progressive, relentless damage, death occurs tragically early, usually by age 2 or 3.
Let's just quickly touch on that molecular insight again, the protein folding issue. You mentioned this discovery is actually fueling modern therapeutic research.
Yes, absolutely. Molecular biologists found that many of the mutations causing Tay-Sachs don't actually stop the hexa protein from being produced altogether. Instead, they cause it to misfold. It doesn't achieve the right 3D shape. And this misfolded protein gets flagged by the cell's quality control system, something called the unfolded protein response, and it gets destroyed prematurely, usually in the ER, before it even has a chance to get to the lysosome. This insight was really the spark that ignited the whole field of molecular chaperone therapy. The idea is if we could somehow stabilize that unstable, misfolded protein, even just a little bit, maybe we could rescue enough enzyme activity to slow down or even halt the disease. It's a crucial concept we'll definitely come back to when we talk treatments.
Okay, fascinating. Moving on then, let's talk about Niemann-Pick disease, NPD. This is actually a group of disorders, types A, B, and C. It sounds crucial to distinguish between A and B, which are enzyme problems, and C, which is different.
That's right. Niemann-Pick types A and B are true enzymatic deficiencies. They're caused by an inherited lack of the enzyme sphingomyelase. This failure leads to the accumulation of its substrate, sphingomyelin, but also secondarily cholesterol within cells. And like Tay-Sachs, types A and B are notably more common in Ashkenazi Jewish populations. The clinical difference between A and B really comes down to severity. Type A is the severe, classic infantile form. It involves extensive neurological damage, rapid progression, and sadly, early death, usually within the first few years of life. Type B is generally much milder. Patients primarily suffer from massive enlargement of organs like the spleen and liver organomegaly, but typically have little to no central nervous system involvement. This allows them to survive into adulthood.
What's the characteristic look pathologically for Niemann-Pick A and B? What do the cells look like?
Well, because sphingomyelin is broken down in those MPS cells, the hallmark finding is lipid-laden phagocytic cells, macrophages that we call foam cells. These foam cells accumulate in the liver, spleen, bone marrow, lymph nodes, basically anywhere you find macrophages. And these cells become enormous, sometimes up to 90 micrometers in diameter. Their cytoplasm is filled with countless small, uniform lipid vacuoles, which gives them that striking, foamy appearance under the microscope. Under the electron microscope, you see different inclusions compared to Tay-Sachs. Here, you see structures known as zebra bodies. These are described as concentric, lamellated myelin figures. Looks a bit like a zebra's stripes, hence the name.
Zebra bodies, okay, got it. And Niemann-Pick type C is completely different at the molecular level, you said, not an enzyme deficiency.
Correct. Type C is mechanistically distinct, and interestingly, it's actually more common than types A and B put together. The defect here isn't in the enzyme itself, but in non-enzymatic lipid transport out of the lysosome. It's caused by mutations in either the NPC1 or NPC2 genes. These genes code for proteins that are essential for moving free cholesterol and some other lipids out of the lysosome and into the cytoplasm where they can be used. When these NPC proteins fail, the free cholesterol essentially gets trapped inside the lysosome, and you also get a secondary buildup of gangliosides there too.
So given that unique defect, a transport problem, not a breakdown problem, what's the clinical picture for type C?
It's known for being highly variable, notoriously heterogeneous. This can make diagnosis quite challenging. It can present anywhere from a severe form in newborns, even fetal hydrops, to a much slower adult-onset neurological decline. Typically, though, it presents as a chronic neurological disorder. Key features often include difficulty with coordinated movements, that's ataxia, maybe slurred speech, and a very specific, almost diagnostic sign, vertical supranuclear gaze palsy. That's the inability to voluntarily move the eyes upward or downward properly. All this leads to progressive psychomotor regression. And that link we mentioned earlier, type C increasing Alzheimer's risk, really underscores how essential proper lysosomal cholesterol transport is for maintaining brain health and preventing protein aggregation over the long term.
Right. Okay. Really complex. Finally, in this lipid group, we come to Gaucher disease. You mentioned this is the most common LSD overall quantitatively, and the one with that really strong proven link to Parkinson's disease.
That's correct. Gaucher disease results from a deficiency of the enzyme glucocerebrosidase, sometimes called GCase. Its job is to cleave the glucose residue off a lipid called ceramide. When GCase is deficient, its substrate, glucocerebroside, accumulates massively, primarily within phagocytic cells, the macrophages. And again, yes, it's notably common in the Ashkenazi Jewish population.
Well, we definitely need to define the clinical subtypes clearly here. There are 3, but type I is the vast majority of cases, right?
Yes. Type I is the chronic, non-neuronopathic form. It accounts for about 99% of all Gaucher cases. And crucially, in this type, the storage of glucocerebroside is limited to the mononuclear phagocytes. The central nervous system is spared. Clinically, these patients typically present with massive splenomegaly. The spleen can become huge, sometimes 10 times its normal weight, causing pain and discomfort and also hepatomegaly, liver enlargement. Accumulation of Gaucher cells in the bone marrow is also a major problem. It causes bone erosion, bone pain, increased risk of fractures, what we call pathologic fractures. This causes significant morbidity, but because the brain isn't involved, patients can often live a relatively long life, especially with treatment. Then you have type 2 and type 3, which are the neuronopathic forms. Type 2 is the acute infantile form. Here, there's almost no detectable enzyme activity. It leads to rapid progressive CNS involvement, and sadly, death usually occurs within the first 2 years. Type 3 is intermediate. Patients have the systemic involvement, the enlarged spleen and liver, the bone disease, but they also have progressive CNS disease, which usually starts to emerge in childhood or adolescence.
And the pathology of the Gaucher cell itself is really unique looking, isn't it? It's described as looking like crumpled tissue paper, which is such a vivid image. It's not foamy like a Niemann-Pick.
It is absolutely unique, and pathologists rely heavily on that distinction. The accumulated leukocerebrosides arrange themselves within the macrophage cytoplasm in a way that gives it this distinct fibrillary appearance. It's often likened to crumpled silk or, as you said, tissue paper. It's definitely not the small, clear, uniform vacuoles you see in foam cells from other lipid storage diseases. It's a key diagnostic feature. And we absolutely must emphasize two critical points about type I Gaucher. First, the big spleen and the bone destruction aren't just due to the physical storage of lipid. The accumulating glucocerebrosides actually activate the macrophages. These activated macrophages then secrete inflammatory cytokines, things like interleukin-1 and TNF-alpha, and these cytokines actively contribute to the bone erosion and cause systemic inflammation.
So it's an active inflammatory process driven by the storage.
Exactly. And second, that Parkinson's connection is absolutely key. It's not just patients with full-blown Gaucher disease who are at risk. Even carriers, people with just one faulty copy of the GCase gene, who have reduced but still present enzyme activity and usually no Gaucher symptoms themselves, even they have a significantly elevated risk for developing Parkinson's disease later in life.
And the proposed mechanism ties right back into that vicious cycle of impaired autophagy we talked about earlier.
Perfectly. The reduced GCase enzyme activity impairs the overall efficiency of the lysosome. This leads to impaired autophagy, and that in turn prevents the efficient clearance of misfolded proteins like alpha-synuclein. So alpha-synuclein starts to aggregate. There's strong evidence now for a kind of reciprocal negative relationship. Low GCase activity seems to lead to higher alpha-synuclein aggregation, and conversely, aggregated alpha-synuclein appears to further impair GCase activity. It creates this detrimental feedback loop that likely fuels neurodegeneration. This connection is precisely why Gaucher disease research is arguably at the forefront of developing new therapeutic strategies for Parkinson's. Understanding the LSD is unlocking pathways relevant to millions.
That's a phenomenal insight and a perfect segue into our next section. We need to shift focus now from lipids to other crucial macromolecules that can accumulate. Okay, let's shift gears now and talk about the mucopolysaccharidoses, or MPSs. Here, the failure is in breaking down different kinds of molecules, glycosaminoglycans, or GIRCs.
That's right. GAGs, which used to be called mucopolysaccharides, are large, complex sugar molecules. Things like domidin sulfate, heparin sulfate, keratin sulfate. They're really abundant components of connective tissue, the excellular matrix, cartilage, basically the glue holding tissues together. The MPSs are a whole group of related, generally progressive disorders. They result from deficiencies in the specific enzymes needed to break down these different GAGs step by step. When these GAGs aren't broken down properly, they accumulate inside lysosomes. And this affects pretty much all the cell types that are responsible for producing and maintaining connective tissues. So you get accumulation in MPS cells, those macrophages again, but also in endothelial cells lining blood vessels, and in fibroblasts throughout the body. The common sites where you see severe involvement reflect this distribution: the spleen, liver, blood vessels, heart valves, joints, and unfortunately often the brain as well. Microscopically, the accumulation creates what pathologists call balloon cells. The cytoplasm appears cleared out and highly vacuolated because it's stuffed with these water-soluble GAGs.
And this widespread accumulation in connective tissues gives rise to a very characteristic sort of generalized set of clinical features, quite distinct from the lipid storage diseases we just discussed.
It really does. The buildup in connective tissue throughout the body leads to what are described as coarse facial features. Also, progressive joint stiffness is very common, making movement difficult. And often, there's intellectual disability due to CNS involvement. The JAG deposits in the cornea of the eye frequently cause corneal clouding, which leads to progressive vision loss. That's a key feature in many MPS types. And importantly, cardiovascular complications are a major issue across this category. You get severe deposits building up in the coronary arteries and on the heart valves. This is actually a very frequent cause of premature death in many MPS disorders.
Okay, let's maybe focus on the two main examples people might have heard of: Hurler and Hunter syndromes.
Right. Hurler syndrome, which is technically MPS type IH, is generally considered the most severe form. It results from a deficiency of the enzyme alpha-hydroxylase. It follows the typical autosomal recessive inheritance pattern. Clinically, it's characterized by marked hepatosplenomegaly, severely stunted growth, the coarse facial features, skeletal deformities, and usually profound intellectual disability. And because of those aggressive cardiovascular deposits, children with Hurler syndrome typically succumb to cardiorespiratory failure quite young, often between the ages of 6 and 10 years. Then there's Hunter syndrome, or MPS type 2. Clinically, it can look somewhat similar to Hurler, but there are two absolutely critical distinctions for listeners to remember. First, Hunter syndrome is the key exception to the usual LSD inheritance rule. It is inherited as an X-linked recessive trait. This means it predominantly affects males, as they only have one X chromosome.
Ah, right, the X-linked exception.
Exactly. And second, it generally follows a milder clinical course than Hurler syndrome. And very specifically, the JAG accumulation is usually absent in the cornea. So these patients typically lack the corneal clouding that's so prominent in Hurler. That's a key distinguishing feature.
Okay, X-linked and no corneal clouding for Hunter. Got it. Now, our final prototype disease takes us into what sounds like a totally different category, glycogen storage, but you're saying it's technically also an LSD, Pompe disease.
That's correct. Pompe disease, also known as type 2 glycogenosis, is indeed classified as a glycogen storage disease based on what accumulates. But it definitely earns its spot in the LSD category because the specific enzyme defect is located within the lysosome. The defect is a deficiency of lysosomal acid alpha-glucosidase. It's sometimes also called acid maltase.
So why is the specific location of this enzyme being in the lysosome so critical for classifying it here? Glycogen's broken down elsewhere too, isn't it?
It is, but the lysosomal pathway seems to be crucial for the normal turnover and recycling of glycogen. When this specific lysosomal enzyme is missing, you get massive, widespread storage of glycogen, but specifically inside the lysosomes. This happens in virtually all organs. The glycogen accumulates, distends the lysosomes, and severely disrupts the normal cellular architecture. This is particularly damaging in tissues with high energy demands and glycogen turnover, like muscle. Clinically, the infantile form of Pompe disease is devastating. The single most prominent and defining feature is massive cardiomegaly and enormously enlarged heart. The heart muscle fibers, the myocardial cells, become absolutely engorged with glycogen-filled lysosomes. Under the microscope, this gives them a distinctive lacy or ballooned appearance. This rapidly leads to severe heart muscle dysfunction and cardiorespiratory failure. Tragically, death usually occurs within the first 2 years of life in the classic infantile form. However, this is actually one LSD where enzyme replacement therapy has shown significant success. It can be life-saving, especially if it started very early, before irreversible damage occurs.
Wow. Okay, so whether it's lipids or complex sugars like GAGs or even just glycogen stored in the wrong place, the sheer destructive power that comes from a single missing enzyme within that lysosomal system is it's the overwhelming takeaway here, isn't it?
Absolutely. It highlights the critical housekeeping role of the lysosome. #tag implications, treatments, and modern connections advancements.
We've covered a lot of ground, really delving into the complexity of these cellular failures.
Yeah.
From, tiny errors in mannose-6-phosphate tagging all the way to massive organ enlargement, bone erosion, and devastating neurological collapse. Now let's turn to the exciting part, maybe the hopeful part, how the very sophistication of these diseases, our understanding of them, has driven equally unique and sophisticated approaches to treatment. So what does this complexity mean for the future of therapy?
Yeah, the therapeutic landscape for LSDs is incredibly dynamic. It's constantly evolving, partly because, as we've discussed, the target tissue, especially the brain, is exceptionally difficult to reach with many conventional therapies. We can broadly categorize the current and emerging approaches into, say, 3 main pharmacological strategies, plus the more definitive, potentially curative options. The first, and perhaps the most established approach where it's applicable, is enzyme replacement therapy, or EART. The concept is straightforward. You synthesize the missing functional enzyme in the lab, usually using recombinant DNA technology, and then you administer it to the patient, typically intravenously. And it is undeniably effective, sometimes dramatically so, for the systemic, non-neuropathic forms of these diseases. The classic example is type I Gaucher disease. The enzyme gets taken up effectively by the target cells, the macrophages, which are circulating and accessible via the bloodstream.
Right, but the huge looming challenge remains the blood-brain barrier, doesn't it? If the disease has significant neurological involvement, ERT is essentially useless for the brain component, correct?
Precisely. That's the major limitation. These large enzyme molecules, being proteins, simply cannot cross the very tight junctions of the blood-brain barrier, the BBB. So while ERT can be life-changing, even curative in terms of the systemic symptoms for type I Gaucher, it's completely ineffective for treating the neurological damage in neuronopathic type 2 or 3 Gaucher, or for diseases like Tay-Sachs where the primary pathology is neurological. This fundamental limitation forces researchers to think smarter, to look for solutions that don't rely on getting large proteins into the brain.
Okay, so that brings us to the next strategy, which sounds like a clever metabolic workaround, almost like starving the disease by cutting off its supply chain.
That's a great way to put it. That's substrate reduction therapy, or SRT. The premise here is quite ingenious, and it acknowledges that often the enzyme deficiency is profound, maybe almost total. So instead of trying to replace the missing enzyme, SRT uses small molecule drug inhibitors to specifically reduce the amount of the complex substrate that the body produces in the first place. The logic is, if you can slow down the rate at which the substrate is being made, then maybe the small amount of residual activity from the patient's faulty enzyme, or even just alternate clearance pathways, might be sufficient to cope. It prevents the toxic accumulation from overwhelming the system.
So you lower the burden on the already weakened system.
Exactly. And this approach has shown effectiveness in certain conditions, including some forms of Gaucher disease and Niemann-Pick type C, where small molecule drugs can partially inhibit the synthesis pathways.
Okay. And the third pharmacological strategy, the one we touched on with Tay-Sachs,
Yeah.
that feels like the most elegant, maybe the most sophisticated approach because it tackles the core problem of the protein itself not working right.
Yes, that's molecular chaperone therapy. This is a really highly sophisticated strategy, and it's typically reserved for disorders where the underlying genetic mutation doesn't completely abolish the enzyme protein, but instead causes it to misfold or be unstable. As we discussed with Tay-Sachs, these misfolded proteins often get recognized by the cell's quality control systems and are degraded prematurely in the ER, never reaching the lysosome where they're needed. Molecular chaperones are typically small molecules. Interestingly, they are often competitive inhibitors of the enzyme, but used at very low concentrations. They're designed to bind specifically, but temporarily, to the mutant enzyme protein as it's folding. By binding, they act like a scaffold or a folding template, helping to stabilize the protein structure just enough. This stabilization prevents it from being flagged for destruction in the ER. This allows more of the mutant protein to escape degradation, traffic correctly to the lysosome, and perform its function, even if that function is somewhat reduced compared to the normal enzyme.
So it's like giving the faulty protein a little bit of help to get folded correctly and get to the right place.
Precisely. And crucially, because these chaperones are small molecules, they often have a much better chance of crossing the blood-brain barrier compared to large enzyme proteins. This makes them a major area of hope for treating the severe neuronopathic LSDs that are currently intractable to ERS.
It's really about optimizing the function of the defective protein the patient already has, rather than trying to introduce a whole new one from the outside.
Exactly. It's a form of precision medicine, potentially tailored to the specific effects of certain mutations on protein folding and stability. Now, beyond these pharmacological strategies that aim to manage the disease, we have the approaches that aim for a definitive cure. That's stem cell transplantation and gene therapy. Hematopoietic stem cell transplantation, HSCT, or bone marrow transplant, can be curative for certain LSDs, most notably type I Gaucher disease. This works because the primary disease cells involved the mononuclear phagocytes, macrophages, are derived from hematopoietic stem cells in the bone marrow. So if you replace the patient's bone marrow with healthy donor marrow, you provide a permanent, lifelong source of healthy stem cells that will differentiate into functional, enzyme-producing macrophages, effectively correcting the systemic disease.
In gene therapy, that represents the ultimate goal, presumably, fixing the problem at its genetic root.
It really does. That's the holy grail for many genetic diseases, including LSDs. There's a huge amount of ongoing research investigating various gene transfer methods. The goal is to introduce a functional copy of the missing or faulty gene into the patient's own cells. This might involve modifying the patient's own hematopoietic stem cells ex vivo, outside the body, and then transplanting them back, or potentially using viral vectors designed to deliver the correct gene directly into target tissues, ideally including the CNS. While still highly experimental and facing significant challenges for many LSDs, particularly regarding safe and effective delivery to the brain, the ultimate aim is a one-time treatment that provides a permanent correction for these devastating genetic defects. The whole field is undeniably moving rapidly from just replacing enzymes towards true genetic correction.
The interconnectedness here is it's truly astounding when you step back. These diseases, which individually might seem rare, are absolutely not just medical curiosities. They're fundamental models for understanding how cells fail, particularly in the context of common age-related diseases.
Absolutely. The study of LSDs has provided direct, undeniable molecular links between the failure of the lysosome and the pathology of widespread neurodegenerative conditions. We see the clear evidence: the accumulation of alpha-synuclein in Gaucher disease directly connecting it to Parkinson's, the heightened Alzheimer's risk associated with the cholesterol transport defect in Niemann-Pick type C. It demonstrates that the fundamental failure of the lysosome, this central hub of cellular quality control, has massive downstream consequences. It impairs mitophagy, leading to mitochondrial dysfunction and oxidative stress. It fuels the aggregation of toxic proteins like amyloid and tau and synuclein. These are the very hallmarks of the neurodegenerative diseases affecting millions worldwide. So understanding the consequences of a single gene error in a rare LSD allows us to dissect and potentially target the downstream molecular pathways that are relevant to the much more common, complex neurodegenerative processes.
We've definitely covered a huge amount of territory today. We've gone from the tiny architectural failure of that mannose-6-phosphate tag, all the way to massive organ enlargement, painful bone erosion, and complete neurological collapse. We've detailed the distinct pathologies of lipid storage in Tay-Sachs, Niemann-Pick, and Gaucher, the connective tissue devastation seen in the MPS disorders like Hurler and Hunter, and the unique cardiomegaly driven by glycogen storage in Pompe disease, and the thread connecting them all, that single initial point of failure within the cell's crucial waste disposal and recycling system, the lysosome.
Yes, and if I were to offer a final summary, it's this. LSDs showcase, perhaps more profoundly than any other group of diseases, how an inherited deficiency in a single enzyme, often one that might seem clinically minor or even silent in a carrier, a heterozygote can eventually cascade into devastating multisystem failure and severe pathology. It just underscores the incredibly delicate, intricate balance required to maintain cellular homeostasis. The three main categories of defects we focused on, the sphingolipidoses, lipid storage, the mucopolysaccharidoses, JAGA storage, and lysosomal glycogen storage, pump. They all converge on that central point of failure, the lysosome.
Absolutely. And that leads us perfectly to our final provocative thought for you, our listeners, to carry forward. Given everything we've discussed today, given that LSD research has so clearly illuminated these direct molecular links between lysosomal dysfunction and common neurodegenerative diseases like Parkinson's and Alzheimer's, could it be possible? Could future therapies, perhaps specifically those sophisticated molecular chaperones, originally designed to stabilize rare misfolded enzymes in LSDs, could they eventually become a standard preventative treatment for the millions facing age-related neurological decline? Think about that possibility, using insights from rare diseases to potentially prevent common ones as you continue your own deep dive into the world around you.